The human acellular vessel technology basically involves the use of human bank cells to engineer human arteries from scratch, and then through a process rendering them non-immunogenic so they can be implanted into people. Humacyte has developed a process in which we take banked human vascular smooth muscle cells and then transfer them onto a biodegradable tubular mesh scaffold. This construct is then placed into a bioreactor and grows for 2 months. Over that time, those cells build an extracellular matrix as the mesh degrades. Then, when we use a combination of detergents and salt solutions, we're able to gently remove those cells, creating a human acellular vessel that is quite strong and is ready to be implanted immediately. This technology platform allows us not only to create different vessels in different sizes and lengths, but it also may allow us to create more complex tissue and maybe one day even organs. And what's even more exciting is the fact that we at Humacyte have already developed a commercialization step to this process where we can actually create these blood vessels on a more large-scale basis as opposed to one at a time. Right now, the most advanced application of this technology is our 6 millimeter diameter human acellular vessel. And after about 8 to 9 years of clinical experience in over 450 patients, we have reached some sort of conclusions that are quite interesting. What we've seen so far is the HAV is basically immune from any kind of clinical rejection. When these vessels have been studied, we can actually see that the patients repopulate these blood vessels with their own cells and therefore are very highly resistant to any sort of infection. The additional advantage of creating an acellular vessel that is essentially non-living is that it can reside on a shelf for up to 18 months. And the ability to have an acellular vessel that is readily available off the shelf also is potentially revolutionizing the field of vascular as well as cardiovascular repair. So we're very encouraged by some of our initial results, both preclinically and clinically, in a larger diameter 6 millimeter human acellular vessel. And now what we would like to do is transfer that knowledge and test our smaller 3 millimeter vessel for design in applications towards treating cardiovascular disease.